Hi, I'm Emma.
I'm Caroline.
And I'm Brianna.
And today we're going to be presenting phytoplankton's reactions to ocean acidification.
What is ocean acidification? Ocean acidification is when carbon dioxide is absorbed by the oceans and turns into carbonic acid. This acid is what lowers the pH levels and increases the CO2 in the waters.
And so one type of organisms who may be extremely affected by ocean acidification are phytoplankton. And phytoplankton are a group of extremely evolutionarily diverse single-celled organisms that spend their entire lives floating around on the ocean's currents and tides. And phytoplankton are photosynthesizers, and they're also primary producers, so they're the building block of all aquatic food chains. And think of them kind of like the grass of the ocean. That's kind of their job. And additionally, through photosynthesis, they take carbon out of the atmosphere and into their bodies, and then when those phytoplankton either die or are eaten, part of the carbon in their bodies moves down to the bottom of the ocean into carbon sinks. And then, how about everyone in the audience right now take one big deep breath? I need one too. And so 50 to 80% of the oxygen that you just breathed in was produced by phytoplankton, which is really amazing.
So even though phytoplankton play a positive role on Earth, there are some species that are actually harmful to both humans and marine life. These are called HAB species, or harmful algal blooms. These species produce a range of toxins so when there's a high concentration of these species in an environment, they can get into the aquatic food chains. And so when they are eaten by, let's say, a shellfish, when humans digest the infected shellfish, they can experience effects ranging from an upset stomach to even death. So a species in our experiment that we used is Alexandrium fundiense, which is actually a harmful algal bloom in the Gulf of Maine.
And so for our experiment, we wanted to research how increasing carbon levels affects both the abundance and diversity of phytoplankton species found in the Gulf of Maine.
We hypothesized that if phytoplankton species are adapted to current levels of carbon dioxide and then introduced to an environment with higher carbon levels, all of the species will experience a population decrease, except for the HAB species, which will have an increase.
So our first step to begin our experiment, we needed to get the phytoplankton. So on our excursion to Maine Maritime Academy, they let us use a ship, which was a research vessel called Friendship, and they had an oceanographic instrument called a CTD, which is right here, and that lets you get ocean water from any depth that you want. So we got a surface sample to be our in-field community sample. And then we also got 2 cultures from Bigelow Laboratories, which was Alexandrium fundiense and the Lactocyrus myspolgii. And the Alexandrium, like Emma said, was a harmful algal bloom, but also the Lactocyrus myspolgii was a non-harmful diatomic.
And so our next step was to filter out our field community sample to get out any zooplankton or other organisms that we didn't want to study. And we also used a microscope to count the abundance of phytoplankton that we found in each of our experimental samples.
We then combined nutrients to the phytoplankton cultures with ethyl group 2 and to the field community.
And then for our experimental samples, we used a yeast culture, which produced carbon dioxide, to bubble into all of our experimental flasks and increase the carbon levels in those flasks, which increases the pH, excuse me.
So throughout our experiment, we were calculating the pH and making sure that our experimental flasks had the same pH level, so we can have our conclusion be that all of the flasks had the same conditions happening to them. And we also calculate diversity using Simpson's diversity. And as you can see on this scale of the pH, we had ours at about a 5 throughout the experiment for our experimental flasks, which is the equivalent to, imagine, black coffee, which is extreme levels for the phytoplankton to be put under, but it was interesting to see how they would react.
And we also kept our control samples at about a 7, which is kind of the same as tap water.
So this is our Alexandria fundiense culture graph. And as you can see, there is a 91.26% decrease in our control and an 89.53% decrease in our experimental. As you can see, on days 7 and 9, the population abundance actually stopped decreasing and started to increase by 116.5%. We think that this increase was in part caused by because since the Alexandria fundiense cultures were exposed to a new environment, it might have taken them a couple of days to adjust to the new levels of nutrients put into them.
So the other culture that we decided to study was Thalassia visflogii. And over the course of the experimental sample decreased by 88%. And over the first few days, as you can see, the control sample seemed to be going in the same direction. It was decreasing fairly rapidly. However, between November 2nd and November 4th, it actually started to increase. And between those 2 days, the population increased by 115%. So in total, our control population increased by 8%. And that's really interesting to compare with our experimental sample, which continued to decrease.
So when we put the two graphs together to see how they fared compared to each other, we saw that the Alexandrian, or the Hab species, it started decreasing as the same level as the Thalassia, but then on day 7, instead of continuing decreasing like the Thalassia, it actually started to increase, which is interesting to see because that supports our hypothesis that the Hab species would fare better than non-harmful algae. Also, we calculated diversity throughout our experiment to see the evenness and abundance through our in-field community sample. And as you can see, on days 7 to 9, there was an increase in diversity. That does not mean that there is an increase in abundance of our species, but only that it was more even and throughout.
So some of the species, some of the species that we found in our whole community sample were Melocyra, Didyllum brightwelli, Leptocylindrus, and Thalassia visflogii.
We also saw Alexandrium fundiense, Poricentrum, and Caris socialis.
One interesting result that we found, while as other species such as Leptocylindrus and Thalassia visflogii actually decreased throughout the experiment until day 7 when they are completely wiped out population, Melocyra, which is shown in blue, actually fared pretty well throughout both the control and experimental experiments. Additionally, it's important to note that on day 7, the control had 6 individual species, while the experimental actually only had 2. Another interesting result was that on day 7, there was a spike in Alexandria for both populations, for both experimental and control. And this actually supports our hypothesis because we hypothesized that a Hab species would do better in a lower pH level.
So from the data that we collected in our experiment, we were able to conclude that when Alexandrium fundiense is exposed to a increased acidity environment, it actually increases in population. However, Thalassia visflogii doesn't have a strong tolerance for acidic environments. Additionally, through our whole water sample, we were able to see that both Melocyra and Alexandrium fundiense had a higher tolerance to ocean acidification and acidic environments than a majority of the other species that we tested.
So why does this matter? This is important because a lot of the things that we were hypothesizing and finding out in our experiment, effects the diversity of phytoplankton can lead to impacts in our society.
So since phytoplankton are taking in carbon dioxide in order to photosynthesize, a decrease in phytoplankton would lead to more carbon dioxide in the air, which is also could lead to global warming. And since phytoplankton photosynthesize, they're making oxygen. So the decrease of phytoplankton, that can also lead to a decrease in oxygen.
Additionally, a decrease in the abundance and diversity, or both, in global phytoplankton populations would pose immense consequences on aquatic food systems and the various industries that rely on them. So we would like to say a huge thank you to our phytoplankton master, Dr. Carrie Whitaker. Thank you for helping us keep our experiment alive.
Also, a big thanks to Maine Maritime Academy for letting us use their research vessels to get our whole water sample.
And also a thank you to Bigelow Laboratories for providing us with Blastiosaur Weisfelogia cultures and our Alexandria Fundiense cultures. And next we'll be having microplastics, which is Rebecca, Kira, and Kevin. But before that,
Any questions?
Any questions?
Merivy, is that
With these two cultures that you had, they are the ones that seem to survive the best. What effect or what effect is that likely to have? And will this reduce the diversity as acidification increases because some of the other species did not fare as well?
Definitely. So as we studied diversity, we actually found that through the experiment, the diversity in both of our samples decreased as the experiment went on, and that's extremely important. In our graph, oh, thank you, Brianna. Do you want to go to the diversity graph? Sure. So on day 9, the diversity for the experimental sample seems to be higher. However, there were only 2 species in that sample, while there were 6 in the control sample, and it got a higher score for Simpson's diversity complex index, because it had more even numbers. So it's a little misleading because it looks like there's more diversity and different species in the experimental. However, by that point in the experiment, a majority of the species had died off.
And that's why they're missing from some of the later colored graphs.
Other questions?
Well, I have a comment. A direct relationship to what your research is doing is the California crab industry is facing cancellation for this season because of the high toxicity and the poison right now of sea mammals, decrease in birth rate of the California seal pups, and which eat a lot of the crab. So, is this important? Yes.
Other questions? Yeah.
How did you know, when you're out in the vessel and you're collecting samples, how did you know where to go and the depths that you needed to go to collect the samples? I know in high concentrations you can actually see plankton, but I'm assuming under bright daylight, you couldn't actually see it, so how did you know?
Our field community sample is actually a surface sample from the Penobscot Bay.
So phytoplankton are, like we said, they're photosynthesizers, so we would expect them to be on the surface because they're trying to get the sunlight to make the food for themselves.
But you said you needed the harmful algal blooms and the non.
Mm-hmm.
Yeah, to get those.
So how did you
Sorry, go ahead.
that's fine. So how did you, is it so prevalent that you could just pretty much dip a bucket anywhere and get everything you needed? And then you had to split it out? I understand that, you had to find.
So for Alexandrium fundiense and Thalassiosauria, those were actually cultures that we bought. So we just got only Alexandrium in a little vial. However, for our field community species, we just went into the Penobscot Bay, took 2 liters of surface water, and then had to go into the lab and identify all of the species that we found there.
Thanks.
Yes.
You chose pH 5, which is really extremely low pH. 2 questions. 1, why pH 5? And 2, would you expect to see sort of a less extreme response as the pH lowered? My first question is why pH 5?
Sure. So, to be completely honest, when we started our experiment, we had trouble finding a solid number for how much the acidity in the world's oceans were increasing each year. So we thought that 5 was a really great number because it's a really high acidity, and we thought it would be really interesting to see how the phytoplankton species reacted to such an extreme environment.
Did you do any other studies along the way between 8 and 5, or just 5 and compare to 8?
We chose a solid 5. I would love to do another study where we slowly increased the acidity throughout the experiment, but that was really difficult for us to do within the parameters that we had.
Thank you.
All right, any other questions? All right, student.